So our next talk, we're going to be switching tribes here, going from the less woody to the more woody. And this is going to focus a little bit more on a population level talk. So Eduardo, you want to give us some insights into your work?
Present this study leading by my former student Luis Perez-García and all the female team that with all this analysis. So first, I want to introduce with the bamboo word, talking about the phylogenetic. So we can see here Bambusoideae is sister to Poaceae, we see in the talk with the Paul. So it's and the sister clade is Orizoideae. More or less, is the Poaceae is classifying 12 different subfamilies and more or less 800 genera. Are I missing something? Yeah, more or less, and 12,000 species, more or less, and increasing the numbers with the news, the new things that is coming in the next months. So the Bambusoideae is divided or is classified into three tribes, right? It's a Bambusae with two clades, the paleotropical guri bambus and the neotropical guri bambus. And sister to these two is a Olire based on chloroplast markers, actually, or plastomes. And the tribe, the temperate guri bambus arundinaria. Guaduinae is sister to Artrosolidine, so I want to talk about a little bit more Guaduinae. That is, the species are in this subtribe. So more or less are around 1,700 described species right now, but will be more soon. So Guaduinae is comprise only 6 genera. Apoclada and Hemocalon are sister genera, and Guadua is sister to this one, to these two genera based on chloroplast. Otatea and Olmeca form a clade, and sister to all this is Tibicia, is an Indian, what is in Cuba, another island species. So, and sister to Guaduinae, of course, is Artrosolidine subtribe. So Guaduinae is no es so like a rich subtribe like other ones, but it's some kind of diverse in Mexico, for example, and in the Amazon region. And Jimena Londoño has been working with this subtribe for several years, describing many new taxa, more of them from Guadua species. And the numbers are these: Apocladis, only 1 species; Eremocaulum, 5 species, maybe will be less. Guadua, 35, will be more. Olmeca is only 5 species. Otatea, 13 species, and will be more, like 20 someday. Tibicia, only 2 species, and in total, 61 species in this subtribe. So it's so a small one. It's not so rich like the other ones. And there is no color. So the distribution of all the Guaduinae species going from Mexico to Argentina and Uruguay. So you can see here, and in this square is in Mexico, where you can see at least 4, 5, 6 species from Guaduinae. You can see Guaduas, Otatea, and Olmeca in that particular region. So it's not so rich like the other subtribe. And Costa Rica is not the main part to see Guaduinae species. It's interesting. You can see here a lot of Chusqueas, other species. So in this unpublished phylogeny using RATSEC, you can see here the 2 species that we did analysis. Guada velutina is inside of the clade Plexifolia, and this clade sister to Guada Angustifolia, clay, and plexifolia, and Guada trinii is sister to all these clades. So in the base, we have the Mexican one, Guadua longifolia, and probably a new one species from this complex group of the species in Mexico. So in Mexico, we have 7 Guadua species: Guadua culeata, Guada plexifolia, Guada inermis that is endemic, Guada longifolia, Guada paniculata, Guada tuxtlensi, and Guada velutina, both are endemic too. And in Argentina, we have Guada chacoensis, Guada paraguayana, Guada taguara, Guada trinii, and Guada variegata. And more or less, is using the GBI data, all the Guadua are here. So you can see here in Colombia, because there are more records from Guadua angustifolia, no more species of Guadua. So some species in Mexico, you can see Guada paniculata, Guadua velutina, and Guadua culeata. This is the tallest bamboo-like tree in Mexico. So here is Jimena, and the base of this big Guada culeata in Veracruz, in Mexico. Some of the species of Guado have been used for the native Mexican for so long time. So they use these species to build the houses. So here we can see Guado paniculata, Guado plexifolia, and inside of this wall is Guado velutina. So 3 species of bamboos have been used for bareque or bajereque technique, that is put mud between the walls and then cover with the other stuff, and you have a house, beautiful house. And of course, Guada angustifolia is the species more used in Latin America, because the strength of this species. And with this species, you can build these beautiful things that Simon Vélez did in Colombia. So talking about flowering is the process where it's the only process where the bamboos exchange gene flow between the populations, right? So, but this is not like a normal plant like the other ones. So the bamboos has asynchronous or synchronous flowering pattern, have more or less long flower cycles, and then monocarpy. I mean all the plants that most of the plants die after flowering. So in this context, Guada trinii is a mass flowering plant, and the cycle is more or less 30 years. That is important for the study. And Guada velutina is a sporadic flowering species, and the cycle is more or less 30. So more or less all the Guada is around 30 years between each flowering cycle. So Guada velutina is distributed is the species with the northernmost distribution in Mexico, well, in the continent. Here is San Luis Potosí, Tamaulipas, Veracruz, and Puebla. And it's growing around, it's an riparian species with a asynchronous flowering. And it's around by sugarcane, corn, citric, and coffee crops and grasslands. So the plant is living around the rivers, but outside of the rivers is this. You can see all this. There is no any forest along right now. Guada trinii, Yatebo or Tacuara brava, this is the common name in Argentina. They grow in Brazil, Paraguay, Uruguay, and of course Argentina. This species is a riparian species with synchronous mass flowering, and they use the culms to build or to make these instrumental music instruments. So what is the aim of this? The study was to elucidate the spatial patterns and influencing the genetic diversity and the structure of the 2 American species of Guada velutina and Guada trinii, and evaluate the significances of the environmental factors for these 2 species. So to do that, we collected for Guada velutina 11 populations, 169 individuals, and using 8 nuclear macrosatellites. So you can see here more or less the data, the records from the elevation. around here in the San Luis Potosí is like 100 kilometers like the between the populations, the southern and northern population. So for Guada trinii, Lia Monti collected 9 populations, 143 individuals, and 9 nuclear microsatellites. You can see here the elevation of the population. Most the population, like this one, are inside of the Parque Nacional Iguazú, and these other populations are outside of the park. So the results for Guada velutina, you can see here, the main results is the genetic variation is more or less low, and the genetic diversity is low, and the structure is more or less a little bit high. And the structure analysis says that there is more like 5 different genetic groups. So you can see here, that color belongs to a group of populations. So but that there is a lot of gene flow between the populations. And the FT, you can see here populations that are more that does not share so much gene flow between them, because it's are populations that are pretty far, like a naranjo with population in the south. So it's a normal one for a species that have a long distribution. And using this analysis, you can see here that there is a pattern of isolation by distance, because it's a little bit long, and more or less like a three or two groups of genetic different groups. And doing different analysis to see what the variables are that explain more this pattern, we found that the rivers actually are the variable that is important for the connectivity for the populations. So the rivers is pretty important for the gene flow between the population when the plant is flowering. So the conclusions are low genetic diversity in this species, at least 5 different genetic groups, high genetic structure, and the river acting as a corridor for gene flow for this species. So what happened with Guada trinii? Guada trinii is the genetic variation is higher than Guada velutina, but the structure is not. And only we found 2 genetic different groups. So you can see here the populations with the sharing some genotypes between them, between each population. So the genetic the comparisons is more or less high in some population, but not for all. So this population is in this part, and for example the PS is other one. So there is no isolation by distance in this species, like the other one. And the grouping is more or less like a 1, maybe 2 or 3, 4 different genetic groups. And the analysis with similar to the other one, to the Guada velutina, is different here. Here, the connectivity is explained by the rivers, the precipitation of the wettest month, and the elevation are the main variables that explain this connectivity, this pattern that we found. So it's important for the species, these variables, ecological variables. And doing a different analysis using the ecological niche model, all the samples that we download from GBI in blue is the best predictor for the niche for the success for the species. So our collection was more or less here. So using this information in a different analysis, we found that the populations are connected. So the population is healthy, or not the species, the population that we use for this analysis are in the best place where the plant could live for survive. So there is no any how do you say, any danger to because it's a healthy, or there is a lot of gene flow between the populations. So the conclusion is this species has high genetic diversity, low genetic structure, rivers precipitation and water month and elevation contribute to gene flow, and we found spatial environmental connectivity. So that is important for the health for the species. Talking about something like that, like similar to health. So comparing our results with previous one, here we can see that Guada trinii has a higher genetic diversity compared with Guada velutina, and more or less similar to Guada angustifolia. And yet the genetic structure is higher in Guada velutina, but not so high like in Guadua inermis. Guadua inermis is the other one endemic for Mexico that has also sporadic flowering pattern. So there is you never saw the whole population flowering, and there is a few individuals per population. So that is important. I don't say that, but Guada velutina and Guada trinii has big populations. I have a lot of time. Thank you. There is population that have thousands of individuals or hundreds of individuals. So Guadua inermis only you can find like a 20, 30 individuals per population. So and the other one is a big one, a population, so are healthy in some way. So and having big population is better for the gene flow and to maintain the diversity, the genetic diversity for the species. So the last one, I have a lot of time to questions. I want to thank Luz, my former student, Yessica Alquicira, Yessica Rico, Ofelia Vargas, and Monti from Argentina. And thank you to all of you.
We have time for questions.
So what could you hypothesize what's the explanation for the rivers? Why are the rivers driving or correlating with that genetic diversity?
If we can see when the plants are flowering in the rear, there is some kind of gene flow. So the temperature is different from the rear, from the top to the below, and if there is no like an wind, maybe the gene flow is more or less regular. But you can see the gene flow through the seeds, to the fruits, is going to the river down. I think that could be the explanation why the river is important.
Another? Okay. Another.
We have a lot of time for questions.
Since we have the time, I'm gonna follow up. But so how did you look at the rivers? Were they?
Mm-hmm.
Did you break them up, or what? How did you connect the rivers to the diversity?
Yeah, that was difficult for Luz. We use the base of the rivers, and she need to connect the rivers for taking that information like an variable. So that process is a little bit complex. So is the way that, because we have different, in San Luis Potosí we have different rivers, and there is no connected actually. But you need to connect all the rivers through the to the basin more or less, and to see how the river variable is acting in the gene flow. Mm-hmm.
Another one? Okay, thank you very much.
You're welcome.